continue. Sleep is for the weak. It's true, Avash. I agree wholeheartedly. That's a great way to start this recording. Hi, this is the overview for preparing for the dog paper. I am your, oh, I don't like that. I want to see everybody. I am going to be your tutor this evening, morning, midday. God help us all. And we're going to be going through my little write-up to-do list for making your dog paper. I just finished grading your fly paper, and the improvements overall. But for some people, you didn't take my comments to heart last time, and you should have. So I'm going to be repeating myself a lot in this from the previous video, but I also want to emphasize the other PowerPoint presentation that I made for the in preparation for the last paper on how to write an introduction, which very much applies to, or how to write a background, which very much applies to the introduction here. All right, so I will repeat again to all the new people arriving that this is recording. If you would like, for the people in the room, feel free to join us on Zoom if you want to share your screen, if you want to see my screen share, or you can just crowd behind me and look at it. I'm as low tech as possible in this current day and age. Let's get to it. Step one, you're going to have to do some prep work. I recommend doing this yesterday. The first step is to read the instructions. I will show you where to find that in a minute, and we'll go through it for the Canvas tour. But you're going to read the explicit instructions that are available to you, and then you're going to make a list of all of the documents that you would like to have on hand while you write this thing. In that list must be dog data document, which I'll show you in the Canvas tour, the rubric for this course, for this event, for this paper, whatever, that I'll show you in the Canvas tour, the dog lab handout and worksheet, which you should have a copy of from Canvas, but also that worksheet finished. Hopefully you can reach out to your lab members if you don't have a picture of that, and make sure that you do, because you're going to need it. And you also are going to want to easily access the lecture notes specifically for that week where we talked about dog stuff. So let's go on a quick Canvas tour. Stop share. Boop. Boop, 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 boop. Okay. So this is the Canvas page where you go to modules. You collapse all, you can easily scroll down to dog snip report. It's at the very bottom. You click on that and you have all of your resources. This is the instructions. You want that. This is the rubric. You want that. This is the data. You want that. Make sure that they're downloaded. You're also going to want this. It's the example title page for the dog report. Download it, have it. I also heavily recommend watching these 3 videos just to get yourself in the right mind space, even if you feel like you already know all the information. So stop the share. we're actually going to change what we're sharing briefly. I'm just going to do this. So now everyone can see my screen. This is the wrong document. Why are there so many things? I hate it all. What is going on? Aha. I probably will want that though. All right, dog report instructions. Admit. Dog report instructions. This is your best friend. Follow it excruciatingly detailedly with all the details, on all the details. Do it. Have it open. This is the rubric. At a glance, we can see that 50% of this mark, i.e. 10% of your grade in this course, a letter grade's worth, is the content, but 50% of it is the quality of writing. And we're gonna go over how to point mine that. But I also wanna direct you to that earlier document that I posted about how to write an introduction. And that document is found here, where we go home. Where is it? There it is. Office hour information, getting help. Me, useful files, Abi made. Click. There it is, useful files, Abi made. Fly eye paper, video, how to write a background. This thing here is very useful. Read it. Stuff and things. That's all. Okay, so let's go back to a PowerPoint presentation. Once you've accessed all of your supporting documents, read the instructions again and identify the things you don't know how to do or don't know what they mean. And then learn those things. Start with the lecture notes and links in the modules. If you end up using the internet, I recommend starting with Wikipedia. And then verifying things on other sources. Make sure that you verify multiple on multiple places to make sure that whoever is randomly editing that website is not an idiot. Then you write an outline. Don't skip this. I know you want to. Don't start at the top. Write an outline. Use the instructions and the content part of the rubric to build an items list of all of the things you need to write, like a to-do list with checkboxes. And write in the corner what points will be awarded to each element so that you can think about how important it is to do each thing and prioritize, and don't spend too much time on something that's only worth a half a point. Then you take that item list, you insert all of the relevant info that you have gathered from your various resources, including and especially the dog lab handout from lab 12 and the dog data document from Canvas. And you insert those into your items list. You can even screenshot pieces and copy and paste. This is just kind of a storyboard of what we're doing. We're going to do a special methods outline and a special introduction outline. So just include headings for those. We're going to do special methods and introduction outlines. So just include headings there. Then you go back through the instructions and rubric and make sure you didn't miss anything. Make sure that every point is accounted for and every topic, every to-do list topic has a descripting name, a title. Yeah.
So when you say going through the points, you mean just going through the content, right? Not the quality.
Yes. Yes. I mean, specifically, this is the content part of the rubric. We're going to focus on that first. Good point. And I'm color coding things loosely such that things that you make are blue, things that you haven't made yet and need to make are green, and things that someone else has made are purple. Make sure they're organized in the order that they're mentioned in the introductions, in the instructions or rubric. All right. Methods. She mentioned in the instructions that you can assemble a word bank. In general in life, when a professor says you can do a thing, that means you should do a thing. Do that thing. So you're going to assemble a word bank. I love vocab words. I'm really disappointed that I didn't have a good opportunity for you guys to lose points for not learning vocab words. I would have loved to make you all do what's called a vocabulary project, where you assemble vocab words into a paragraph in reference to each other every single week and get credit for that. But there's so much going on in this course. But I'm a real stickler for vocab words. So yeah, word bank. Doing this first will ensure that you know the meaning of the words before you try to use them. Read through the dog lab handout and list all the words that you learned in this course or in Bisc302. Try your hand at defining them first, then look them up online. Use multiple credible resources to verify accuracy. If you want to use someone else's image to illustrate, that's great, cite them. Do not copy and paste definitions. I have been told to check. I will be copying and pasting them into Google with quotes and seeing if anything comes up. So write them in your own words. I will be able to tell if you don't know what you're saying. I'm very good at this game. Next step is to disassemble the dog lab handout into a painfully detailed outline. And then use that outline to write your methods. So that dog lab handout was the one where you use the instructions to do all the things. Those instructions, translate them into instructions you are writing, or explanation, really. And keep in mind that the introductions will be will have kind of elements of a general methods vibe. This is your detailed methods, if you remember from that very first paper. So details, but also no conclusions. Don't tell me what happened. I don't want to know yet. Just say what you're doing. If you find yourself described, oh, and every point mentioned in the content part of the rubric should have its own heading. Better more headings than less headings. When in doubt, headings. You need figures. She mentions in the instructions, a figure would be really helpful. That means it's required. Do it. If you find yourself describing the way something looks, it needs to be a figure. If you find yourself, if you would draw a diagram of something in order to describe it to someone else, that needs to be a figure. If you leave it out, I will be confused. Okay. I'm going to bold you need figures. That's useful. Okay. Results. Do this after the methods. Don't do it after the introduction. The introduction is something that you write last. Methods, results, then the introduction, or then other stuff and then introduction. Introduction's last. Results. She's very specific about this. So I've in red here copied and pasted from the instructions. She says, briefly give the genotype of the dog for all the SNPs we could identify. Translation, say simply and clearly in a couple sentences, what is written about the dog in the dog data document. Your data is just right there. She gave it to you. Just rephrase it. Just say what it is at the top of the page, at the top of the results. Point out the ones that you couldn't identify and briefly suggest reasons for this. Translation, in a couple sentences, say which SNPs we tested for, but don't have the results for, and why we don't have the results for that. Next, tell the reader all the expected PCR product sizes and cut and uncut sizes, then point out which SNP variants are associated with which traits. Jesus, that sounds like a terrible paragraph. Don't write that paragraph. Translation, with your words, refer to a table. You can also describe what is in the table here, but don't write an impenetrable list of SNPs. A table might be the most effective way to present this information. A well-designed table could provide all this information together very efficiently. This is code for a table is not optional. Make a table. You'll probably lose points if you don't include a table. Don't say in the results, your dog has blank SNP, which is why they have blank phenotype. That's for a different section. Instead say, your dog has blank SNP, which is associated with blank trait. We're not talking about any specific dog here. These are we're not talking about like analysis. These are just objective truths of the results. Any questions about the results?
I have a question. When you said that explain why we couldn't.
Oh no. I cut out for a second. Say it. I can't hear you.
Can you hear me now?
Now I can, yes. Explain
Can you hear me now?
Yes.
Yeah, so I'm saying that there's a section where it says that on the results, we have to explain why if certain SNPs didn't work, explain why they didn't work, right? Are we supposed to do it on the results section or on the interpretation section that, oh, why specifically didn't work?
The results section, yeah. So the results section, this point out the ones we could not identify and suggest reasons for this. The reasons are procedural things like we didn't see a band. This might be because of such and such. Or we saw a band, but it didn't digest. This might be because of such and such. So all of those very technical nitty-gritty reasons, that's in the results, and that gives the reader a context for why we're not going to be talking about it in the interpretation. Yes.
So we only have information, like, as far as I'm aware, we only have like data on what like the genotype is, not like pictures of the gels. So we wouldn't be like, I don't think we'd be able to say like there's a band here, but it's not. You know what I mean?
Yeah, I do.
We would just say in general, these are why we might not see results.
That's what I would say, but hold on, let me look. Because, oh, that's less helpful. I will, I think that we say that, like, you know, that might be because of this.
Like just generally speaking.
Generally speaking, this gel didn't, this SNP didn't work for whatever, for these are reasons why it might not have worked. But I will double check with you. And the answer to that question will be here. I'm going to just make this all red so that I remember to check with Kathleen. Okay. That. So I remember to check with Kathleen about that question, because I've had that question before and I don't actually know the full answer to it. So, any more questions before we move on? Everybody good back there? Awesome. All right, interpretation. I didn't write a lot about this because there are pretty good instructions in the instructions, but this is where you say how the SNPs make sense or don't, given what we know about this dog. What we know about this dog is a single picture of the dog, and there are some identifying things in the handout of the results. Follow the instructions closely and you will rely heavily on the Lab 12 worksheet that you filled out with your lab group. Let's look at the instructions for that, eh? So this is your interpretation instructions. I think it's pretty clear. Follow those instructions. I'm not going to read them out loud to you. You know how to read. I believe in you. This is also, if you note in the rubric, just one giant lump of 8. Interpret the results. Group SNPs associated with snout length together and the same associated with size. So you're going to group them together. You're going to say what the results were for this dog and thus what you would predict the phenotype for this dog to be and whether or not that's accurate. And if it isn't accurate, potential reasons why it might not be accurate. Does that make sense?
I'm sorry. Can you hear me?
Yes.
What is that exact worksheet you are talking about? Can you just show me again?
Yeah. We go to modules. Data for the dogs in the study. I think this is what you're talking about. I don't seem to have downloaded it. Just open it up.
No, the other worksheet you showed us that we did as a group.
Oh, the
is-
worksheet we did as a group.
Is that the lab handout or is that the one you gave us back?
It's the one you gave us back. We gave you back.
Can you just show me again because
I don't
don't-
have it. Does anyone-
okay.
Yeah, because I have it's the worksheet that you filled out with your group. If you don't have it, reach out to your group member who kept your collective group work. They should make sure that everyone has it.
Okay.
They should have they should take pictures of it and send it to you. Does that make sense?
Yeah.
Yes. Those.
Supposed to.
Are you comfortable showing it?
Yeah, sure.
It's this
stuff.
thing.
And this stuff.
Make sense?
Yes, thank you.
Yeah, wonderful. Okay. Moving on. Uh-huh.
It says, did the dog sample help to confirm our hypothesis? Is that just that our hypothesis is that these SNPs are associated with these traits, and does that dog have those SNPs and those traits?
Yes. Yes. The question was, when it says, does this data help confirm our hypothesis? The hypothesis is that these SNPs are predictive of these traits. So if the dog has the traits that we think are associated with these SNPs, then it confirms our hypothesis. And if the dog doesn't have those traits, then it doesn't confirm our hypothesis. And we should suggest alternative reasons why. Like, maybe it's a polygenic trait. Maybe it's, maybe the dog's a mutt. Make sense? Cool.
Sorry, could you repeat the hypothesis once again?
Hypothesis. I'm going to write it down. Hypothesis. These SNPs are associated with these traits. If the dog has the traits, the traits predicted by the SNPs, by its SNPs, then that would support our hypothesis, period. Make sense? Thank you for asking to clarify. That's very valuable. Positive reinforcement. Let's talk about the introduction. This is the most important part, I think personally. The skills of writing a good introduction will serve you well in literally anything you want to do in the future, even if it is to write a scathing blog post about the guy who drove your Uber yesterday. Like anything that you write, if you want to be convincing, you need to be able to organize your story so that people care about the details you're about to give them. So the first step is to go back through your methods and interpretation and list all the concepts that need to be connected. The point of an introduction is to connect all of the things so that they make sense in the context of each other. You want your reader to see the heading RFLPs caps and think, oh yes, that's how they're going to blah, blah, blah. I know why this is relevant and I care. They know why because you told them why. You told them in the introduction. Use the instructions and rubric to organize the list into an outline. This list. The list will be green. There we go. The first sentence, this is from the instructions. The first sentence is a general statement of your research goals. We are looking at dog DNA to see what SNPs are associated with snout length and size, I think are the things that we're looking at. I don't remember, whatever they are. The last sentence is a specific statement of the goals that apply to this dog. We are going to use the DNA you sent us to look at the SNPs at these locations from your dog and see if those SNPs are predictive of your dog's phenotype and fit with our larger data set. Everything else goes in between. Kathleen wrote this instruction section. There's also other stuff in the rubric, and in general, anything that you mention in the methods and in interpretation that needs to be connected to the goals of the overall project. All of that fits between these two things and carries the reader from the general concept of we are looking at SNPs and associating them with phenotype to we are going to do this specific test with your dog to see if it fits into our larger data set. And those things include dogs are cool, huh? And what is an RFLP? Does that make sense? Do we mention coat length in the intro? If you're going to mention coat length later on in the paper, yes. Did we get SNPs about coat length for this dog? No? Then I wouldn't bother. I wouldn't mention coat length. Does that make sense? Yeah. Okay, cool. Unless, of course, you're using it as some sort of like example of like previously it has been determined. Now we're looking at this other thing, which you could do. Okay. After you've done that, you've made an outline. Isn't that wonderful? Now you're going to go back through this outline and make sure that everything mentioned in the rubric and the introduction has a place and a home in this outline. Now you can write your introduction. And the outline is a checkbox list. It's a to-do list. You want it in the order that it's supposed to be in. You want it each paragraph needs to have its own thing it's talking about. The first sentence of a paragraph is, this is the thing I'm going to talk about. The last sentence of the paragraph is, that ties into this next thing I'm going to talk about. do we not mention we looked into the SNPs for coat length in methods then? Did we look?
So CS2 is a coat length.
Okay.
No one got coat length, but none of the twos worked.
Oh, fascinating. Yeah, totally. Yeah.
So we should mention.
You should mention that. Yeah, you should say, we're looking at these things, including coat length. And then in the methods, you say, we're going to do these SNPs. And in your little table in the results, these are the SNPs that are associated with coat length in our larger group. And then in the analysis, we say, or was it in the, yeah, in the results, right? In the results, we say, we didn't get any information for coat length. Here's a potential reason why. Sound good? Thank you, Simran, for clarifying. That's very helpful. Good questions. Okay, are there any questions about the content and procedure of writing an introduction?
I have a question.
Yes, please.
So when you went over this, so after we write it, you want us to write an outline before we write our introduction.
You
Right.
write an outline for everything, and then you write your methods, and then you write your results,
Oh my gosh.
and then you write your interpretation, and then you write an outline about what goes into your introduction. Okay? Okay. Is it possible for us to is it possible for us to go over how to interpret a SNP result from a dog? Stick around for after 4, Cameron, and we can go over that in more detail. But I'm not going to include that in this recording. This is just focused on how to write this paper. Okay. Okay, so we're going to move on to writing quality, which is 10% of your course grade and half of this thing. And I want to emphasize again that how to write a background presentation. It is very good because it talks about the structure of a paragraph and a sentence. These are things that, especially as native English speakers, I say to the native English speakers, I am bad at, which is I make these sentences that are long and convoluted and they make internal sense to me. But that's because I have my brain and I'm just assuming that everyone else has my brain. They don't, I don't. So I say this to everybody, but, and obviously pay attention to it non-native English speakers. Keeping your sentences simple is helpful for translation purposes. It's helpful for like being clear, but especially for native English speakers, keep your sentences simple. Don't say actually, although, interestingly. It's not necessary, and you might accidentally say something factually incorrect by saying although and then saying something that isn't an although. Don't do it, just say no. When in doubt, be boring and write. Boring and write. Write short, simple, repetitive sentences. Skip modifiers. Don't say alternatively. Don't say, it is a curious fact that. Just say the thing. Be boring. Look at this like a jigsaw puzzle, not an essay. Everything fits together to create a united whole with a predetermined structure. You are not an artist. You're just sticking the bits together. There are a lot of them, and they all have a place where they go. And the goal is to make the picture as clear as possible. Yes, I will be on here until 6. Someone asked, can I come back much later to talk about interpretation of the SNPs? And yes, I will be here until 6 with the Zoom room open. Okay, presentation matters. There's a whole section about presentation here. Upload a .pdf, not a .docx. When you upload a .docx, all of the formatting of your figures goes out the window, and Canvas smooshes it and morphs it into a horrible monstrosity. And then we have to email you, and we're already tired and irritated when we get to grading you. So it's not that I'm trying to be extra mean to those people. I don't think I am, but please be nice to me so that I can be nice to you. I'm in your corner. But print to PDF before you upload these documents. Oh my God. Okay, thanks. That's all. That was my rant there. Follow the cover page template. The cover page template. Title page for dog report. It's right here. Ah, I pressed the wrong button. Oh, there it is. What? Oh, no, fine. Okay. This thing. No, it's not this thing. It's thinking very hard. This thing. You're just going to open this file. You're going to write the name of your dog here. You're going to write your name here. You're going to write your student name here. And you're going to write the date here. And then you're going to write the rest of your paper in this document so that the format of this page is unchanged and pristine. Sound good? Unchanged, pristine. Don't get fancy with me. No additions, no creativity. Just use that template. In general, with presentation, add a heading wherever possible. While you're writing, it will make it easier to read. For me to read it. Just add headings where it makes sense. Be consistent with the formatting of your headings. Make sure everything is in the same font. Make sure that if you're going to do italices or bolds or anything, it's consistent. Your systems are consistent. Capitalize the first letter of every sentence. That I can't, there's so many of those, you guys. Just proofread, please. Okay. And my final big suggestion here, before we go to the next page, is read it out loud to a stuffed animal or a mirror or whatever with your voice. Out loud. Especially if I've told you before in your feedback that your sentence structure is sometimes confusing. Read it. No excuses. With your voice. Because it will make more, you'll realize where the grammar is broken by saying it out loud. And even if you're English second language, you'll get that in your head. You'll feel it. Because it'll be a little bit clumsier. And if you're not sure, be boring. If you're not sure if the sentence makes sense, make it shorter and 2 sentences. Last step. And this is the most important step. I can't emphasize this enough. Sleep on it. Even if you're already running a day late, sleep on it. And in the morning, eat food, drink coffee, and then, or tea or whatever you do in the morning. Then read the instructions and the rubric again, and then read the paper out loud again and grade yourself against the rubric. Go through your checklists. Make sure that you didn't miss anything. Edit as you go for clarity and completeness, and then turn it in. I cannot emphasize this enough. It's worth being 2 days late and catching all the typos. You will get more marks because you remembered at the very last minute to include a description of why the DNA separates in the gel or whatever it is. You will remember, you will read it and you'll be like, oh shoot, I mentioned this thing in the methods. And I added this thing later in the methods and I didn't add it in the introduction to, I need to go back and do that thing. You'll catch it on the next day. Trust me, it's worth it. Okay, that is what I have. Are there any questions? Amazing. I am going to stop my recording. Thank you so much for joining. This recording will definitely go live before I go to bed tonight.